Welcome to the week 2 recording of the Teaching with Data cohort. Today we're going to go over the topic of graphing, and in particular, the rules and the art of graphing. So first, in case you haven't seen any of the earlier recordings, here's a little bit of context for how we're working through this cohort. We're trying to cover the 4 skills of a data scientist. The first is organizing data. And if you haven't seen our video that Aaron and I made about tidy data, I highly recommend you check it out on YouTube. It's a great way to help the rules of tidy data really sink in. Next skill is analyzing data. This is knowing what statistical tests to use and getting comfortable handling those analyses. Communicating data, so taking the results of your analyses and effectively communicating them to your audience. And finally, developing data intuition. And I'll explain a little bit more about what we mean by data intuition on our next slide here. So the normal progression of these 3 skills plays out just like you think they would. You got to organize the data you collect, then you analyze it to see what relationships there are, and finally you communicate your results. And when you go through this process a number of times through different inquiries and experiments, you begin to develop data intuition. And this is really related to each step of that process. And as a student's data intuition grows, they'll become better at deferring to tidy data format and maybe even collecting their results in a neater manner. Then they may become quicker at determining what statistical test to use, or even just quicker in computing it. And finally, they'll get better at following the rules and adhering to the art of graphing. But data intuition is something really that plays out at each step and is developed through a number of iterations going through this progression. Today, like I said, we're focusing on communicating. So quickly, we're going to revisit tidy data. Again, if you want a more in-depth explanation, please check out our blog or the YouTube channel where there's a good tidy data explanation. So in tidy data, the distinguishing factor is that each variable has its own column. And what's important about this is that it requires us to explicitly recognize what variables we're working with, which is, of course, an important consideration when coming up with what visualizations work. Next is that each observation of a variable has its own row. And this could be useful, for instance, when time is one of your variables, because I think we tend as humans to just group our data based on time. But if there are multiple observations being made at the same time, each of those observations is going to require a different row. And then finally, each value has its own cell. Ultimately, tidy data is a really important way for us to recognize our variables and also keep the results discrete so that they can be uploaded and analyzed using different computer software. Now, when it comes to graphing, I think a useful heuristic is to think of the rules of graphing and the art of graphing. The rules of graphing are straightforward. It's a yes/no checklist. You can be either correct or incorrect. First, you need to format your data by thinking in terms of variables. That's why tidy data is important. Second, you have to select your visualization based on your research question and your variables. There are correct and incorrect visualizations based on your variable type and what type of research question you're trying to answer. But the more subtle piece of this is taking what you can create when you've played by the rules and colored inside the lines and really focused on communicating effectively. So these 4 are not necessarily linear. They're criteria that you might want to revisit and tweak as you work on your graph. First, consider using color effectively. It might relate to your real life variables. Use spacing in a way that's pleasing. So again, things like jitter, the placement of descriptive statistics on the graph, all have a visual element on how your data is being received. You don't want data points to be too crowded, for instance, because that will block an important message that the distribution is telling your viewers. Next, use statistics that best represent the distribution of your data. Know when to use mean and median-based descriptive statistics. And finally, keep your audience in mind. You're going to want your graph to look a little bit different, or visualization to look a little bit different if it's appearing as a part of a science fair poster or if it's being inlaid in a research paper. To get a little bit of practice with the rules and the art of graphing, we're going to consider an experiment that Aaron did with his high school classrooms while he was in the Chicago public schools. They sought to answer the research questions: do female brown anoles lizards prefer wet soil when looking to make a nest? And do female brown anoles lizards prefer to nest near plants? So again, it's very important that we keep those questions in mind because it will determine what visualizations we're able to use when going through the rules of graphing. So here's a quick description of what the experimental design looked like. You can see the 2 variables that are being considered are the presence or absence of a plant, as well as 5 different categories or types of soil moisture. Here's an actual look at the 10 separate pots of soil, those with plant and without, and then their aquariums. Here are some brown and old lizards featured in the experiment. And then let's get to it. So first we're going to consider the rules of graphing, and we're going to go to DataClassroom to do this. So the data set we're looking at has already been properly formatted, and all we need to do is consider our research question and then select the best visualization. And we're going to use the graph wizard to do this. Now, what's great about the graph wizard is that it helps students who may not yet be comfortable selecting graphs, who haven't developed their data intuition, to really reduce their decision-making to process of elimination, as long as they keep their research question firmly in mind. So if we look at these 5 different ways to determine what type of graph we're making, we can say proportions. Okay, that seems like what we might be working with. We know it's not change over time. We know it's not variability, but it kind of is comparing groups, and it's kind of a relationship. So let's recall those questions specifically. Do female brown-nosed lizards prefer to nest with plants or without? So that variable is plant or no plant. That's a categorical variable. And the second question related to soil moisture. We had 5 different categories or types of soil moisture. So we are looking at data distributed among different categories and not 2 groups of numerical measurements or a relationship between 2 numeric variables. So we can eliminate those 2. And as we just covered, we are looking at 2 variables. So we're going to select this. And now what are our 2 variables? They are soil moisture and plant presence. And I'd like to start off with a bar graph because everyone likes to start with bar graphs. Here's a good example of the scaffolding that DataClassroom provides students. To take it step by step for the sake of time, we're going to do all of them and get right to the graph. So here we have a data visualization that we've created by playing by the rules. We've colored within the lines. This is what we've got. And this is a correct graph. But I think we can make it better because upon first impression, we can see pretty clearly it looks like the brown and old lizard, female brown and old lizards, did show a preference for soil moisture of 50% or above. What also strikes me right away is I would think, oh, there might be something here about soil without a plant because of the difference in height. But if I look over to the y-axis, I see that the count here is really 16, and this is 20, which is not that far off. And so I'd like to change the spacing and the color to kind of better reflect what the data are actually telling us. The first thing I'm going to do is change this to a stacked bar chart. And that first impression now kind of leaves me with, okay, the green area and the purple area look a little bit closer. So my first impression isn't, oh, there's a difference there that we should investigate further. One thing I like to do is adjust the color to reflect the actual variables that were being studied. So I'm going to change this teal in the yes to more of a green, and I think I'll change the purple no to something, to a brown, maybe just to represent the soil. So I'm going to go to the values here. Let's pick a color close to brown. There we go. And let's pick a green that's not too fluorescent. All right, there we go. And we haven't really picked an audience, so I don't know how to adjust the size of the labels. And I don't really think we need descriptive statistics here, but let's go ahead and copy this image to the clipboard. And may as well drop it right back in this presentation. Look at that. Just like that, we have an effective graph that showcases not only is there very clearly a presence for soil with higher moisture, but also that the female brown and old lizards didn't seem to care whether there's a plant or not. All right, well, the rules are pretty straightforward. Let's take a little bit of time to practice picking out ways we can utilize the art of graphing. So to quickly revisit them, the rules of graphing: format your data, think in terms of variables, and then select your visualization based on your research question and those variables. The art of graphing: use color when you can, use spacing, use statistics that best represent the distribution of your data. Again, that's mean or median based. And finally, keep your audience in mind. And that could be anything from how you're presenting the paper to the actual age or scientific experience or knowledge of your audience. So our first example here is a data set that has followed the rules of graphing, taking a look at average temperature anomalies over about the past century. And one thing that's immediately displeasing to my eyes is how much overlap there is of the dots in the middle. It also makes me think of just like little hole punches. But we could probably play with the spacing to fix this. And then if there were any more subtle trends, we might want to use a color or a color gradient. And so here's a quick look at a graph that's used both of those really well. We can actually see that this graph math really wasn't playing by the rules because there were variables that weren't properly expressed. But you can see the color has done a great job of highlighting the difference in these temperature anomalies at low latitudes, middle latitudes, and high latitudes. And there's even regression lines fitted to the three different kind of subgroups of data there. And then the key is reducing the dot size so it looks more like a distribution as opposed to a trash heap. All right, let's take a look at another one. This is from our biodiversity lesson where scientists in the Amazon photographed different species of mammals approaching different salt licks. And there's clearly a story here. You can see at licks F, G, and H, there's more animals appearing there, but we can really do our audience a favor by adjusting the spacing and some of the size here. So instead of having to kind of focus and compete to figure out how many different species were visualized at F, G, and H, it's much easier with the stacked bar chart. You can quickly count 4 here, 4 here. And then by adjusting the size and the spacing, it's much easier to read. I've got pretty good vision and I was squinting to read the letters over here. All right, this is from an experiment where scientists were analyzing how running with shoes and running barefoot affected different runners' strides or their cadence. We follow the rules here. We have a graph that's perfectly fine. It looks like you could reasonably conclude that when runners run barefoot, their cadence increases, which you can think intuitively makes sense. They're taking shorter strides because they don't have any shoes on. But when we think about color, we can help tell a much more interesting story, including, most importantly, being able to see right away the two anomalies to this data. So for 2 of the runners, their mean cadence actually decreased, and that's not clear at all in the graph just playing by the rules. So here we have some data from the classic potato osmosis lab, and you can see quite clearly that there is a difference in the mass change for concentrations with low molarity versus high molarity. But when we take into account the descriptive statistics as well as use a little bit of color, we can tell a much clearer story, and we even reduce the spacing as well to get rid of this kind of overlap effect. So it looks like here the color gradient is just used to communicate the increase in molarity. We've got a box and whiskers plot to help better see the spread of these little distributions, and the spacing has made it easier to see the distributions as well. And this is a very professional looking graph versus just what you get by playing by the rules.